Thank you everyone for joining us today for our pre-application webinar. Before introducing our speaker, I want to go over some webinar logistics and describe the goals of the webinar. During this webinar, all lines will be in listen-only mode. You may submit questions at any time using the Q&A or chat panel and select all panelists. And this is the image you see on the left side. If you do not see this box or these panels, you may need to activate the appropriate box by using the floating navigational panel, and this may be found on the center of your screen. And you'll click on the icon that looks like a bubble to expand your chat and Q&A windows. If you need it, closed captioning is available by clicking the link in the chat box. And please note that this webinar is being recorded. So hi, my name is Leah Mechanic. I'm from the Division of Cancer Control and Population Sciences, or DCCPS. And we are one of the divisions that participates in the ITCR program. Our mission in DCCPS is to reduce the risk, incidence, and deaths from cancer, as well as enhance the quality of life from cancer survivors. To address this mission, we conduct and support an integrated program of the highest quality genetic, epidemiological, behavioral, social, applied, and surveillance cancer research. So in our participation in ITCR, we've seen relatively fewer applications coming from our population science community. And you can imagine that there are several different types of tools that may be of interest to population scientists. And I've highlighted some or summarized some here on this slide, and I'll only have time to go through a few bullets on this slide. And these tools will include things like collection tools that support data collection, such as from surveys, wearable technologies, or social media or spatial data, or tools that support the extraction of information from medical records on patient-reported outcomes. These tools can also include those things that combine and help access data, such as tools that support data harmonization across cohorts and populations, or tools that support our cancer registries, symptom registries, or other repositories. They also could be tools in analysis and interpretation, which may help with the integration of different data types, or tools to support population-based modeling of cancer risk, prognosis, or outcomes. And there are other types of tools that I have not listed here. And Julie, in her presentation, will give you some specific examples of population sciences projects that are currently supported by ITCR. So this brings us to the purpose of today's webinar. We want to encourage applications in population sciences to the ITCR program, and we want to share information about the resources available through the ITCR program or tools that you may use in your research. Please note that questions about the scope of the funding opportunities will be addressed during this webinar. However, questions about investigator-specific study aims will not be addressed. And I should add that while this webinar is focused on population sciences, the ITCR program supports research throughout the cancer continuum and research from other divisions. And now I will introduce Julie Clem as our speaker today. Julie Clem is the acting branch chief of the Cancer Informatics branch at the Center for Biomedical Informatics and Information Technology at the National Cancer Institute. And she is the program lead for the ITCR program, and she will go over these funding opportunities and talk in more detail about the program on the next several slides. Thank you, Julie.
Great. Thanks very much, Leah, for that introduction. And it's a pleasure to have the opportunity to speak to this audience about the ITCR program and specifically to raise awareness of the program's resources and funding opportunities with the population sciences research community. So in my talk today, I'm going to start by giving you an overview of the ITCR program, including its history, the overall goals of the program, and the activities we conduct through the program, including the resources we support. After that, I'm going to spend some time walking through the funding opportunities that support the program and walk through the distinctions among the 4 RFAs that support the Informatics Technology Development Goals. This will include the specific requirements of each RFA, as well as general requirements across the program. Finally, I'll talk about some future directions for the program, and we'll then allow time at the end for Q&A. So what is ITCR? ITCR is a trans-NCI program that supports investigator-initiated informatics technology development driven by critical needs in cancer research. The program plays an important role in connecting informatics technology to the research priorities of the NCI. All of the resources developed through ITCR are open source and freely available to academic and not-for-profit users, and most of them are also freely available to for-profit uses as well. We encourage the development of interoperable resources in the program, and we put a particular emphasis on the development of user-friendly resources and the broad dissemination of those resources. Our website, the URL shown on this slide, is a really good resource to you for information about the program, both about the resources available through the program and the funding opportunities I'll be talking about. The program was initially conceived and planned for back in 2011. And at that time, the NCI did not have a single coordinated program to support informatics technology development. Technologies were being developed through individual programs, but these were not coordinated with one another and led to some duplication and gaps. Recognizing this need, the NCI initiated the ITCR program, and the first program announcements were published in 2012. The program was first renewed in 2015, and just this past year, in December 2018, the NCI Board of Scientific Advisors approved the second renewal of the program through RFAs that we are releasing this year and that I'll be talking about today. The technology development supported through ITCR is supported through four funding opportunity announcements that roughly follow the life cycle of informatics technology development. This includes an R21 mechanism that supports algorithm development and other innovative computational research. A UO1 mechanism supports early stage technology development, and that's defined at the prototyping and early hardening stage. We use a U24 mechanism to support advanced stage development of promising informatics technologies to support their enhancement and broader dissemination to the community. And finally, we use another U24 mechanism to sustain resources that are already very widely used in the community, and the community has come to depend on them. And this mechanism supports their continued availability to the community. The funding opportunities have 2 receipt dates per year. The next receipt date will be June 11th of this year, and we'll have a second receipt date in November 20th of this year. These RFAs were approved for 2 reissuances, so they will be reissued in 2020 and 2021, each with 2 more receipt dates. All of the applications received through ITCR across all of the funding mechanisms are reviewed together by an NCI-convened special emphasis panel. Since its inception, ITCR has funded about 80 grants to support informatics technology development, and these are all available for you to see and explore on the ITCR webpage under funded projects. But generally speaking, the portfolio of projects we've supported is summarized in the pie chart on the right side of this slide. And as you can see, about half of the portfolio supports tools and resources that support different types of omics data, including genomics, transcriptomics, epigenomics, proteomics, and several genomic visualization tools. Also, about a quarter of our portfolio supports imaging analysis technologies, and that includes both medical imaging and pathology imaging. We also have a good portfolio of resources and tools that support clinical data and data standards, including the development of new ontologies, as well as technologies that support natural language processing over clinical notes and EHRs. There are resources that support biomarkers and network biology, as well as grants that support protein structure analysis, data transfer technologies, and radiation therapy. So as you can see, the tools that we funded to date have sort of a lower representation in the epidemiology and population sciences space. And as Leah said in her introduction, part of our goal for today is to encourage this community to submit proposals to the program. However, I will note that the resources that we are supporting through the program are available to you and can be applied to your research programs. And in fact, we maintain a catalog of all of the tools that are available for use on our website under the Informatics Tools page. And the records for each tool contain several types of information that are useful for understanding and using the tool. That includes links to the tool website or, as appropriate, a running instance of the tool. It contains links to the source code for these tools. And again, as a reminder, all of the tools developed through the program are open source. They contain links to the NIH reporter record for the grant supporting the tool. And finally, many of the tools provide links to short explainer videos that provide a high-level overview of the tool and its capabilities. The types of informatics technology appropriate to ITCR is very broad. And in the FOAs, as well as on this slide, we provide some examples of the types of technologies appropriate for the program. That includes data acquisition software, data management, storage, and data sharing resources, tools for data mining, visualization, and analytics, data annotation tools, statistical methods, natural language processing, and so forth. So without reading through this whole list, I want to emphasize the point that ITCR supports a broad scope of informatics technologies for cancer research. As I mentioned in my introduction, the ITCR program places particular emphasis on collaboration among investigators in the program, particularly to support collaborations that forward the interoperability of the tools. In order to support interactions among the investigators, we have monthly PI conference calls, annual face-to-face meetings, as well as a number of investigator-led working groups. And the graphic shown on the left of this slide is a depiction of some of the connections that currently exist among ITCR tools. In the graphic, different tools are shown in the gray ovals. The current connections between them are depicted in the orange lines. The connections that are work in progress are shown in the blue lines. And those that investigators have identified as interesting collaboration opportunities are shown in the dotted lines. To help support and enhance these collaboration opportunities, the grants, the U01 and U24 grants, are required to set aside 10% of their annual budget for collaborations that are proposed post-award. We've also used administrative supplements at times to support collaboration opportunities as well. Finally, I'll mention that NCI does support informatics technology outside of ITCR, such as through our R01 programs, and as appropriate, we've invited those investigators to participate in the ITCR program activities in order to benefit from this network of experts in cancer informatics. On the next few slides, I just want to give examples of grants that are funded through each of the four mechanisms in ITCR to give you a sense of some of the ongoing research. So I'll start with an example of an R21 grant, and this is a project led by GQ Zhang from University of Kentucky to develop an ontology-driven faceted query engine for the Kentucky Cancer Registry. And through this grant, Dr. Zhang is proposing to develop a tool called Oncosphere, which would be a query engine that would use the NCI thesaurus as a nested faceted query system toward the goal of providing a web-based exploration tool for the Kentucky Cancer Registry. Now that's the driving use case for this grant, but the long-term goal of the grant is to create data exploration systems for NCI SEER program, as well as other cancer-related data resources through these data science innovations to ultimately transform user experience with a new generation of data interaction modalities. So you can see here in this R21 grant, this investigator is using this funding opportunity to explore new ways of querying cancer data. The next example I want to present is that of a U01 grant, and this is led by Obi Griffith at WashU. Through this grant, Dr. Griffith and colleagues have developed a resource called the Clinical Interpretations of Variants in Cancer, or CIVIC. So CIVIC is a curated web-based knowledge base of the clinical significance of cancer genome alterations. All of the information available through CIVIC is free, and while much of it has been curated by the WashU team, there's also a great deal of information that's been contributed through several major data donations, including those from Illumina and ASCO. The tool has also been adopted by other groups who work on genomic variants, and that includes the ClinGen Somatic Working Group. The CIVIC team has been a particularly collaborative member of the ITCR program, and their tool has been integrated with several other tools in ITCR, including CBio Portal and the UCSC Genome Browser. And I'll also mention that this group has been collaborating with an investigator in one of our sister programs, the Innovative Molecular Analysis Technologies program, to develop data sets that would help drive identification of rare cancer variants. And this is another collaboration opportunity we promote through the ITCR program. The next example I want to share is that of an Advanced Development U24. This grant is led by David Hanauer at the University of Michigan to enhance the Electronic Medical Records Search Engine, or EMERSE tool, which is a tool that extracts phenotypic information from unstructured clinical notes in EHR systems. So EMERSE is designed for broad dissemination and is EHR system neutral, and it's aimed at supporting the ability to integrate data from multiple sites at multiple institutions. And in fact, through this grant, Dr. Hanauer is collaborating with 5 other institutions who are installing EMERSE and creating an informatics research network to establish best practices for the implementation and use of EMERSE across a network of participating institutions. Finally, I want to provide some examples of work that's being funded under our Sustainment U24. As a reminder, the goal of our Sustainment U24 is to support the ongoing availability of highly used cancer informatics resources that the community has come to depend on. And this slide has 3 examples of projects that we're funding through this mechanism. And that includes the Integrative Genomics Viewer, which is supported by Jill Meseroff's group at the University of San Diego. The IGV provides integrative, interactive data visualization of microarray and NGS data at all genomic scales and at large data set sizes, and really has become a de facto standard for genomic viewers within the cancer community, with a very large user and citation statistics. A second example is the CBio Portal for Cancer Genomics, which is supported by Nicholas Schultz's group at MSK, among other collaborators. So CBio Portal is a well-known tool that provides visualization, analysis, and download of large-scale cancer genomics with a large number of unique users per month in the instance supported by MSK, as well as providing multiple other instances to serve specific program needs. A third example of a Sustainment U24 project is the Cancer Imaging Archive, which is NCI's primary resource for acquiring, curating, managing, and distributing images and related data. Again, this is a resource with many thousands of users per month and millions of images with a high level of citations. So with those examples, I would like to next walk you through some specific information about each of the ITCR funding mechanisms and what we're looking for in applications to each of these funding mechanisms. I'll start by talking about the R21 mechanism, again, which is targeted at the development of innovative informatics methods and algorithms for cancer research and management. As a reminder, R21s are intended to support high-risk, high-reward research with little or no preliminary data. So in the research strategy, we ask for applicants to describe the potential of their work to support a transformative capacity for a cancer-relevant area of research. They should also describe the level of innovation of the proposed computational method or algorithm and the advantages it offers over relative to those currently available. So here it's very important to put the work in context of the current state of the field. Finally, and importantly, we ask applicants to describe the level of interaction with collaborators in the cancer research field and the potential for wide adoption by the relevant cancer research community. So here we're asking for you to describe how potential users of this technology will provide input into the work to ensure the utility and usability ultimately by end users. Next, I'll talk about details of the U01 opportunity, which is for early stage development of informatics technologies for cancer research and management. So again, this is for the early development of enabling informatics technologies that address needs across the cancer continuum and supports development from the early prototyping through to hardening and adaptation of the technology. So here the research plan requirements are somewhat more significant than the R21s. We ask that applicants describe the significance of the proposed informatics technology to the collaborating cancer research projects, as well as to cancer research overall. And it's important to provide examples of scientific use cases that would be supported by the technology. I just want to comment on this point of collaborating cancer research projects. Again, it's important that the informatics technology be clearly tied to needs in cancer research, and working with collaborators in the relevant cancer research field is highly encouraged, again, to ensure the utility and usability by the intended users. We also asked the applicants to describe the level of innovation of the technology, again, and putting it in context over competing technologies. The plans and processes for working with the cancer research collaborators to ensure the technology addresses their research needs. And related, the approach for soliciting feedback from end users, again, to assure usability and utility for the research community. Because this is a technology development grant, we want to see the plans and methodology for the technology development, a timeline and milestones for achieving the project goals, as well as plans for participating in ITCR program meetings, including the annual meetings and working groups, as well as the program-wide education and outreach activities. Here, again, emphasizing the importance of collaboration and networking as part of the ITCR program. The U24 Advanced Development funding opportunity has many similar requirements to the U01 that I just described. Again, as a reminder, this U24 is for the advanced development and enhancement of emerging informatics technologies that address needs across the cancer research continuum that have demonstrated potential to have a significant and broader impact. So many of the elements of the research plan are similar to the U01, but I do want to call out an additional requirement. At this stage of development, we ask that the applicant describe their plans for supporting the user community, including things like documentation, training, and other outreach activities. Again, emphasizing the importance of dissemination of the technology to the intended user community. One additional requirement in the Advanced Development U24 is that the applicant provide evidence of the development status of the existing software that's the subject of the application. Again, showing that that this project is really appropriate for the U24 Advanced Development funding opportunity, which means there is existing software that is to be enhanced through this grant. And so examples of the development status include things like a pointer to a running hosted instance of the software, or a mechanism to install and evaluate the software locally, such as binary files, a Docker container, or source code with appropriate instructions, and so forth. So some sort of evidence of the development status of the existing software is required. Finally, the Sustainment U24 has somewhat different requirements for the research strategy, as you might expect, given the distinct goals of this funding opportunity. Again, this U24 is for the continued development and sustainment of high-value informatics research resources. So these are focused on sustaining operations and improving the user experience and availability of these existing, widely adopted informatics tools and resources. So I've highlighted here some important distinctions in the research plan for the Sustainment U24. And that includes providing the demonstrated impact of the informatics technology to cancer research and the justification for why it should be sustained. In addition, the applicant needs to provide plans for evolving the informatics technology to address the emerging needs of the targeted research communities to ensure that it maintains relevance to the research it supports. Finally, I want to note that the Sustainment U24 requires applicants to include a plan for the long-term sustainment of the informatics technology, and this is beyond the period of the award, to encourage investigators to think of ways to support the technology beyond grant funding. I will note that this criteria is not a scored criteria, but is for reviewers to comment on. All four of the ITCR FOAs require the applicants to provide letters of support, but the goals of these letters are somewhat different across the continuum of the four opportunities. In the R21, we require applicants to provide at least 2 letters of support from cancer researchers for whom the proposed method or algorithm would benefit their field of study. In the U01, we also require at least 2 letters of support. These would be from collaborators in the cancer research community. And these letters should provide details on the nature of the proposed collaboration and how this new technology would benefit their research. In the 2 U24 opportunities, the bar is a bit higher in terms of what we want to see for letters of support. So for the Advanced Development U24, we ask for no more than 20 letters from collaborators and or potential users in the cancer research communities who will provide details on the nature of their proposed interaction or collaboration and how the new development will benefit their research. For the sustainment, no more than 20 letters should be provided for how the technology has benefited their research and why it should be sustained. Across all 4 of the ITCR FOAs, we require applicants to provide a software dissemination plan that's aligned with the requirements that are outlined in the FOA, again, to support the open source requirements of ITCR. So the software dissemination plan should describe an approach for making the software freely available to the nonprofit sector. The software should include a license that permits the dissemination and commercialization of the software. The software should be transferable to other teams for development in the case that the current team no longer can or cares to support the software in the long run. The license should include the availability of others to modify the source code, and teams should have a plan to manage and disseminate community code contributions to their source code. I do want to mention that the software dissemination plan is not a scored criteria, but is reviewed and commented on by reviewers. prior to funding, program staff do review the software dissemination plans and may negotiate modifications so that the plans align with these requirements. And these software dissemination plans do represent a commitment by the institution to support and abide by the plan, and the final version of the software sharing plan becomes a condition of the award. All 4 of the ITCR FOAs are clinical trials optional. And this is actually a new feature of the ITCR RFAs. The previous funding opportunities, which were program announcements, were actually clinical trials not allowed. And we made the decision to make the ITCR FOAs clinical trials optional as we prepared for the latest renewal of the program, and we're reviewing the portfolio of informatics technology supported by NCI, and notice that many technologies that are being supported, especially in the DCCPS portfolio, are informatics technologies that are themselves an intervention. And in order to validate these technologies, the validation plan follows the structure of research that could be characterized as a clinical trial by the NIH's new definition of a clinical trial. And for those of you who might not be familiar with the current NIH definition of clinical trial, I've included a link in the slide here, or you can certainly Google NIH clinical trial definition. And this link actually has a very nice and easy to use decision tool to determine whether your study is classified as a clinical trial by the NIH. So I just wanted to give an example of an informatics technology project that includes what NIH would consider a clinical trial in its scope. And this example I've actually taken from an actual grant that NCI has funded, but I've abstracted some of the information to make it more generic. But I did want to share this example so that you can understand what we mean by a clinical trial in this context. So in this example, the investigator is developing a web app technology to improve the implementation of a particular cancer prevention strategy. So the goal of the grant is to create a novel digital intervention that will provide targeted information concerning a particular cancer prevention strategy to a particular at-risk audience. So now we can ask the 4 questions from the NIH clinical trial decision tool to determine whether the validation study included in this project would be categorized as a clinical trial. So the first question is, does the study involve human participants? The answer is yes. It involves a specifically targeted community of participants. 2, are the participants prospectively assigned to an intervention? Again, the answer is yes. In the project, a group randomized pre-test, post-test controlled design is being implemented. 3, is the study designed to evaluate the effect of the intervention on the participants? Again, the answer is yes. The study will test the hypothesis that more people in the intervention group will adopt the strategy than those in the comparison group. And finally, is the effect being evaluated for a health-related biomedical or behavioral outcome? And again, the answer is yes. The study aims to improve the adoption of the prevention strategy to promote reduction in the incidence of this cancer type. So the answer to all 4 of the questions is yes, and therefore this study would be considered a clinical trial and could be proposed in one of the ITCR funding opportunities. Next, I just want to say a few words about the collaborative set-asides that are required in the U01 and U24 funding opportunities. In all 3 of these FOAs, awardees are required to participate in additional collaborative activity projects that are identified post-award. And this is intended to support activities that enhance the utility and or interoperability of the informatics technology they support. So the kinds of collaborations that we envision can be supported through these set-asides include, but aren't limited to, the extension or adaptation of a tool that supports the specific needs of a cancer research project. Technical collaborations such as implementation of a common application programming interface to support data exchange among tools. Or, for example, the adoption of common data standards to support semantic interoperability across resources. Or use of a common software platform or interoperability infrastructure for tool integration. Again, these are just examples of the types of activities that are suitable for support under these collaborative set-aside funds. And so I wanted to provide just 3 examples of set-aside collaborations that have been or are being supported through ITCR. So the first example is a collaboration between OB Griffith, who, as I mentioned earlier, supports the CIVIC tool, with Ian Holmes, who supports a genomic browser called JBrowse. And in their collaboration, they're working to incorporate the cancer variant annotations and protein level features from CIVIC into the JBrowse genome viewer. And this has involved both user interface changes to JBrowse, as well as API changes to CIVIC to allow JBrowse to access the data from the CIVIC knowledge base within the JBrowse viewer. The second example I'll provide is between 2 of our U24 Sustainment grantees that include Jill Meseroff's group, who supports the Integrative Genomics Viewer, and Nicholas Schultz's group at MSK, who supports CBioPortal. And through this set-aside collaboration, the IGV is being enhanced to be integrated into the CBioPortal analysis and visualization environment. And this has involved technical changes to both the IGV and CBioPortal to enable this integration. The third example I'll mention involves a collaboration between David Hanauer, who, as I mentioned, supports EMERSE, and Guilherme Del Fiol and his team, who are supporting the Open Clinical Decision Support tool at the Huntsman Cancer Center. And through this collaboration, they're integrating EMERSE with the Open Clinical Decision Support tools to strengthen Open ODS and its support for family health history data extraction. Finally, I want to say a few words about some future directions for ITCR that are a work in progress. And these future directions were based in large part on our preparation for renewal of the program heading into December of last year. And part of our preparations involved having an outside evaluation performed of the program to provide input into the accomplishments and future directions for the program. The evaluation committee recommended that ITCR continue to support informatics technology development through the mechanisms we had funded through program announcements and that we are now continuing to fund through RFAs. In addition, they suggested we add components to better enable the adoption and implementation of these technologies within the cancer community. And based on those recommendations, we're adding 2 new components to the program. That includes competitive revision opportunities to support the adoption and implementation of ITCR technologies, as well as standing up an ITCR Education Center. So the mission of the ITCR Education Center we envision to be to conduct activities that engage the research and informatics communities to use and extend the ITCR technologies. And a key activity of this new center will be to provide in-depth courses and workshops on topics in cancer informatics that will incorporate ITCR tools as exemplars in these education programs. In addition, this education center will be a center to provide guidance and support to informatics tool developers that will help them facilitate dissemination of best practices for user engagement and outreach around these technologies. We are currently preparing this RFA to be released in early summer of this year. The second component that we're adding, revisions to support the application of informatics technology to cancer research, are intended to address challenges in applying informatics technologies developed through the ITCR program in support of NCI-sponsored research. So these revisions will really be supplements, if you will, to existing NCI grants to support incorporation of ITCR technologies into their research plans. And some examples of activities we imagine would be funded through these revisions include adaptation of one or more existing tools to meet the unique needs of a given research program, or the generation of data necessary to support the appropriate application of the informatics technology. For example, integration of informatics tools to support research goals, or in some cases, system installation and configuration of a system at an adopting institution. And we anticipate releasing these RFAs later this summer. So I hope that's provided you with a helpful overview of the ITCR program. As I mentioned, the website is a very good source of information about the resources developed through the program, as well as information about the funding opportunities themselves and the grants funded to date. I'm always available for questions at this email address, and feel free to call me. And if you're a Twitter user, I encourage you to follow the @NCIDataSci handle, especially the hashtag NCI-ITCR. Thank you very much, and I'll be happy to take questions now.
Great. Thank you, Julie, for a fantastic presentation. And for those of you in the audience, if you have questions, please put them in the Q&A or chat box and send to all panelists, and we'll be able to answer on your behalf. So, Julie, to start, we've got several questions that have come in. One of the questions was about how does one choose between a U01 and U24 mechanism? If you have a tool that was initially developed and deployed, but you want to add new features, is that appropriate for the U24?
Yeah, that's a great question, and one that actually I get frequently from potential applicants. So generally speaking, the enhancement of an existing technology that has demonstrated potential for significant and broader impact would be appropriate for the U24 Advanced Development RFA. However, if you're planning to take the technology in a very new direction, for example, adapting it to a new field, the U01 may be more appropriate. So generally speaking, the U24 is best for enhancing existing technology, but there are times if you're doing something really new with the tool that a U01 may be more appropriate. And if someone has a specific question, feel free to reach out to me offline, and I'm happy to discuss.
Great. A related question is that if you're applying for the U24, must you first apply for a U01?
Yeah, also great question, and the answer is no, you don't. And in fact, most of the U24s funded through ITCR were based on technologies that were developed through other funding sources, either other NIH grants or NSF grants or so forth. So one does not need to proceed orderly through the cycle of grants. I will say, though, now that the program's been running for several years, we're seeing successful U01 investigators now apply successfully and get funding through the U24, and that's something we're starting to see more of as the program has been around a while.
Thank you, Julie. There's been several questions about the collaborative set-aside funds. The first is, is this for work that you're proposing in the parent application?
Yeah, great question, because we use the word collaboration a lot, and collaborations that are central to the aims of your parent grant should be included in your parent grant application and budgeted for explicitly. So you should not assume that you would support those collaborations through the use of your set-aside funds. The collaborative set-aside funds are for collaborative opportunities that are kind of above and beyond your primary goals of the parent grant, and often are opportunities that have been identified post-award. In a way, I like to think of these as built-in administrative supplements. And in fact, we used to exclusively support these collaborations through administrative supplements, but they were so popular and we found they were so important that in the first renewal of the program, we built in these funds into the grants themselves. And now we require that investigators write a short proposal to us going into the second year of their grant, proposing a collaboration opportunity. Those are reviewed by NCI program staff, and once approved, those funds are then approved for use for those collaborative opportunities.
So do the investigators need to identify these collaborators in advance for the use of these funds?
Well, we don't expect these collaborations to be fully fleshed out when you are submitting your parent grant. However, in your parent grant, what you should do is describe your abilities and plans for facilitating future collaborative activities in general. And these plans should include some ideas of possible areas of collaboration that would be appropriate to your project and that complement other program activities. So you want to convey your acknowledgement of the requirement, you're interested in collaborations, and some high-level ideas for what might make a good collaboration. But we don't expect that these other investigators would be on your grant or that those plans be firmly developed when you submit your parent application.
And so related to this, there was a question about for those population or behavioral-based projects, how should they go about collaborating with some of the other ITCR projects that may be more biological or omics based?
Yeah, and I've really been, I think one thing that's been really interesting about the collaborations we've seen through these set-aside funds is one, they sometimes unique collaborations are established that one might not have anticipated pre-award. So I want to say it's great to keep an open mind coming in. That's one point. The second point I'll make is that the collaborations are not restricted to collaborations with other ITCR investigators. I will say that tends to be what happens, but I will emphasize that that's not a requirement. And if there is another tool or resource outside of ITCR for which there's a compelling collaboration opportunity, that would be an appropriate and acceptable proposal for use of your collaborative set-aside funds.
Okay, great. So we did get questions about, what are the areas of interest for ITCR for funding? What topics is ITCR interested in?
Right. So as I showed in the pie chart in my slides, we have a very strong representation currently in genomics and imaging in particular. Those two categories represent about 75% of our portfolio, and that really is a representation of the overall types of applications we receive. However, what we've called out in the current FOAs is that applications from areas that are currently underrepresented in our portfolio are particularly welcome, and that's one of the reasons we're doing this webinar. Is we really want to encourage applications from groups such as population sciences and epidemiology, things like wearables and so forth, data collection from wearables, et cetera. Areas that we have not seen a lot of applications from, we're really interested in supporting and spurring technology development in those areas. We still do welcome, and I know we will get interesting and high quality applications in areas such as genomics and imaging and so forth, because those are very important to cancer research, of course. And those are still welcome, but we're making a special effort to encourage applications in underrepresented areas now.
Great. So I have a question about sort of what responsiveness to the ITCR RFAs and whether or not software as a service is something that would be responsive. And maybe you could talk about generally what people should do if they have questions about whether or not their ideas are responsive.
Yeah, great question. So informatics tool that is utilized as software as a service generally speaking, would be in scope for ITCR. As I tried to outline, we cast a very wide net when we talk about what sorts of informatics technologies we are interested in. And now that we're in a world where cloud computing is becoming more and more prevalent, these sorts of technologies are certainly very appropriate and of interest to the NCI. I will just emphasize our open source requirements that still applies to any software developed through the program. In terms of questions of responsiveness, though, I spend a lot of my time answering questions like that. And if you do have a question about whether your application would be responsive, do feel free to reach out to me. I will take this opportunity to say that a question I often get is, can wet lab work be supported on an ITCR grant? And I want to emphasize that ITCR really is an informatics technology development program. And as such, grants that have any significant amount of data generation are really not in scope for these FOAs. That said, small validation studies that are very central to the testing or validation of the informatics technology can be acceptable if they are on a small scale.
Okay, thanks. We have a couple questions about expectations as far as publishing the tools or posting the tools on the website. So one question was whether submission of your informatics tool to the ITCR website is encouraged or required as a milestone. And then the second related question is about expectations or requirements of the tool being published when applying for any of these mechanisms, or whether or not it's required to be published before submitting proposals.
Okay, great. So the first question is about submitting the tool to the website. So the tool catalog we maintain on the website really is a resource that we, the program staff, provide as one of many ways that we want to help support disseminating information about these tools. And so really the goal of the program is supporting informatics technology development that can be reused by the targeted cancer community. And as such, we post these on the website as one approach to that. So is it required? We've never had anyone say no because generally people want to disseminate their tools. So I'd say yes, it is expected that once it's ready for quote prime time, it would be posted on the website. Then you had a question around the expectations or requirements for a tool being published when applying to any of these mechanisms. So certainly for the R21, the answer is no, because R21 really doesn't require any preliminary data. So that's certainly no for that. For the U01, likewise, this is really for early stage software development. However, it is a U01 and some preliminary data are expected to demonstrate the feasibility of the work. And so we don't expect there to be a tool that's been published, but certainly some sort of evidence of the methods that are going to be established in the technology are expected. For the advanced development U24, as I mentioned, we require evidence of the current state of the existing software. And while a publication of that software is not specifically a requirement, I think it establishing the promise of the software and its suitability for continued development, that a publication can be an important way to establish that there's reason to continue development of the tool. And certainly for the Sustainment U24, there's certainly expectation that there is evidence of broad use of the tool that would certainly involve references or to a publication on that technology.
Regarding dissemination of tools, what's the scope that the applicant has to reach out to? Is it the entire cancer research community, or can it focus on a very specific cancer community that they have already some leverage with?
Yeah, that's a great question, and I think it's really tool dependent. We recognize that some tools or resources, the nature of them are such that they're going to have a really large audience. Something like a CBio portal, for example, is a type of tool that many communities can use and benefit from. And we recognize that other tools have more of a niche focus, and that's certainly a welcome and appropriate type of technology for ITCR. And in those cases, outreach to that targeted community is entirely appropriate. So I think it's very dependent on both the type of technology, the intended community, as well as the state of development of the tool. And I think those will determine what sorts of outreach are appropriate.
Okay, great. So given the breadth of the ITCR program and the many different types of applications that come in, how is review managed, and how do we what is done to just ensure that the breadth of expertise is on the panel?
Yeah, this is a question that we get a lot, because exactly, the ITCR is a very broad-based program covering many domains of cancer research. And this is why we use NCI-convened special emphasis panels to review the ITCR applications. The panel is really stood up after proposals are received for a given round for a given receipt date. And the SRO puts together the panel based on the expertise needed to review the applications that have come in that round. And so using the special emphasis panel approach allows us to bring in specialized expertise that aligns with the type of applications we get in any particular round.
Okay, we have a few questions about the clinical trials. Whether or not, one was whether or not if you answer one question of the 4 that you outlined, it counts as a clinical trial. And the second had to do with whether or not the IRB protocol needs to be in place at the time of review or at the time of application.
All right, so for the first question, do you have to answer yes to all 4 questions? I think the answer is yes. All 4 have to be affirmative for it to be considered a clinical trial. The question about whether the IRB protocol has to be in place is one I would need to follow up on. So I'm happy to, if whoever asked that question could follow up with me offline, I can confirm that with them.
Great, thank you. There was a question about the population-based research emphasis, or what we're discussing here, versus the other tool development RFA for cancer surveillance and epidemiology. Do you want to talk about the distinction between these 2 FOAs?
Right. So I think that I have just a little familiarity with the other FOA that I think was a very specific, targeted opportunity to solicit informatics technology specifically for the SEER data, I believe. And so I can imagine that there could be applications that might be suitable to one or the other. And when it's those cases, I encourage you to reach out to the myself and or the scientific contact on that FOA. And in those cases, sometimes program staff will discuss and make a recommendation as to which FOA might be better for that application.
Great, thank you. If the informatics platform development is already one of the major aims of an ongoing grant, could we also apply to an R21 for additional funding?
I would just say, consistent with NIH rules, you can't have the same work funded through, more than one NIH grant. However, if the work is sufficiently distinct from that being supported through your other grant, then yes, you can apply for that additional work. It just cannot be overlapping with work you're already funding through a different mechanism.
Okay. There was a question about timeline for submission and resubmission. So if an investigator submits an application for the June deadline and it's not successful, will they get the comments in time and be able to resubmit for November?
Yeah, that's a good question, and I will admit that that can be a very tight timeline, getting your comments back and resubmitting. But that is the goal that we shoot for. That one can resubmit to the next deadline. I will say that even though it takes some time for the summary statements to be prepared and distributed, scores do come out quite quickly, usually within a day or two of the review. And from your score, you may have a reasonable idea of whether you'll need to resubmit or not. And so at least based on your score, if it looks like you'll need to resubmit, that's something you can begin working on. And then as soon as you get your summary statements, begin responding to those specific questions and feedback from reviewers.
Okay, great. We had a follow-up question about the use of the collaborative set-aside funds. You mentioned that they would be proposed and reviewed starting in year 2, and how is this review or process for the set-aside funds done? How is it different from the initial grant review?
Yeah, so the initial grant review, of course, goes through the standard peer review process. The collaborative set-asides are reviewed administratively. The set-aside starts in year 2 of the grant. So about 2 months before before year 2 of your grant, you'll need to prepare a short proposal of, outlining what you intend to do with those collaborative funds, what sort of activities you tend to support. It's about a 5-page proposal with the budget sheet and so forth. So that's submitted to myself as well as to the program director. And then we identify staff within NCI who have the appropriate background or knowledge to review the proposal, and that's reviewed by NCI staff. Sometimes we have feedback. We ask for additional information, but then once that's approved, the release of those funds and the approval to use them for the approved project is then indicated in the notice of award going into year 2 of your grant. There are times when the renewal, the non-competitive renewal, comes out before we've had a chance to approve the set-aside, and in that case, we'll just issue a revised notice of award that indicates that the funds are released. So again, generally speaking, the collaborative set-aside proposals are reviewed administratively, and that approval is then indicated in the notice of award.
Great, thank you. There was a question, I think a follow-up question about the dissemination. We're asking whether or not you could say more about the requirement for community-level dissemination with a technological intervention. And there's some concerns about, the required budget to support both the development and the dissemination. And maybe that depends on the mechanism as far as the level of dissemination, but maybe you could talk more about that.
Right. So, we understand that, it's impossible to cover dissemination to every cancer researcher who might benefit from your tool. And so, and I presume, we're primarily talking about the U24 opportunities at this point. And so, we anticipate, the dissemination plans would be targeted to kind of the most promising opportunities for reaching researchers who would benefit from the technologies. So examples include doing workshops at appropriate conferences, holding trainings either through webinars or traveling to key sites to do trainings, providing online materials such as good documentation or good video tutorials. And so really we've seen lots of different approaches to dissemination that are appropriate to the targeted community. And this is probably a good opportunity to mention that one of the investigator working groups that we have in the program that's been one of our more popular groups is one around training and outreach. We have rotating chairs who lead this working group, who bring together investigators particularly focused on sharing their own experiences and learning about best practices from others around opportunities for technology dissemination. And so I think this is an ongoing conversation in the program, is what are the most effective ways to make your community aware of this technology and to support the community using the technology.
So we have just a few more questions. So left, if people still want to ask questions, please enter them in the Q&A and chat box. But maybe Julie, you could talk about the types of projects that may be considered responsive to the R21 funding opportunity.
Yeah, sure. So I'll once again actually just reference the website that we have a funded projects page on the website where you can filter by funding mechanism. And so you can go to the funded project page, filter on the R21s, and you can actually get the list of all of the R21s we've ever funded through the program. And that's a great way to see what has scored well enough for funding. But what you'll see is work that is focused on new methods development and new algorithm development across a variety of research domains, and that's included use of multi-omics data. That's included new approaches to medical image analysis. We just funded a new grant around new calculations for radiation dosage. And I give the example today of a group developing a new kind of query tool focused on cancer registry data. So you can see that it's a pretty diverse portfolio of work, but all focused on trying new methods development in informatics technology.
And you addressed this question in your talk, but maybe you could say more about, what is that threshold for the U24 sustainment? How do we make a decision about what's widely used?
Right. So the examples I gave, I wanted to convey that these are tools that are really very well known in the community. So if you're involved in cancer genomics, you will have heard of IGV and you will have heard of CBioPortal. If you're in cancer imaging, you will have heard of the Cancer Imaging Archive. So I realize that's a little bit subjective, but I think what we're aiming to support are those tools that the community really depends on. And NCI sees a rationale for continuing to sustain and evolve them to meet the needs of the community. We actually get a fairly small number of applications to the Sustainment U24, which actually meets our expectations because it's not a lot of tools that get to that level in their life cycle. So the bar is fairly high there. But we really want to see evidence of widespread use of the technology to be responsive to the Sustainment U24.
Okay, great, thanks. There was a question about the level of funding for the U01s because some of the language in the funding opportunity talks about the NCI's commitment to fund a certain number of awards. Can you just say more about typically what the level of funding is for a U01?
Yeah, so the U01 does have a specific cap that's at no more than 300K direct costs per year for 3 years. And so that's that's sort of hard and fast, and we have a 2.25 million commitment for FY 2020 to support those U01s, and that commitment that's for the fully loaded grant, so that would be to support direct plus indirect costs, and that would support 5 to 6 awards in 1 year.
Okay, great. Thank you, Julie. So I haven't received any additional questions, and I think you've addressed all the questions that have come in, so thank you very much. And so maybe since we're not getting any more questions, maybe we'll go ahead and close out the webinar. So I wanted to say thank you, Julie, for giving us a great presentation today, and thank you everyone for attending. We appreciate your participation in today's webinar. And if you have any further questions about the ITCR program, please feel free to contact Julie Clem, or I'm happy to take some questions myself. And I hope that all of you have a great day. Thank you very much.
Thank you, Leah. Thank you for the opportunity.
All right.